I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
So, cell therapies. Do you know what they are? Can you give me any examples?
Not the scientific aspect of it, but I know that it's very focused on the individual therapies for patients.
It's using engineered cells for treating very specific disease.
Is CRISPR?
Modified T cells.
I have no idea.
Designing cell-specific treatments to, modulate disease systems in the body, kind of tilting more towards individualized kind of therapy.
So it's taking, let's say, the patient's cells out of their body and maybe properly engineering them or modifying them with more specific properties. So maybe like T cells, natural killer cells, re-engineering them so they can be more efficient at their natural function, then delivering that back to the patients to combat different diseases that the patient is experiencing.
Hi everyone, welcome to the show. I'm Danielle Mandikian, and today we're going to be continuing our thread about unmet need. We're going to be exploring an emerging modality, cell therapy, and how it's actually opening up opportunities for treatment. And so I'm over the moon excited to welcome to the bar a bona fide cell therapist, Todd McDevitt. Welcome to the pod.
Thank you. It's great to be here.
All right, so I got a couple questions for you. Okay. First of all, are you a cell therapist? And if so, where do you find all the tiny couches for those cells?
No couches, but definitely I would say that those cells have a lot of anxiety, especially with what we're trying to put them through.
I believe that. All right, so let's take a step back and just kind of do some basics. Cell therapies, what are they?
Really, it's the ambition that cells are another mode of a medicine.
What are their different types? Are they grouped out by function or how they're made?
So an autologous therapy is something that means that they're going to take a cell from an individual, transform it into the medicinal form, give it back to that same individual. And classically, that's been done because of immune rejection. We know this from years of organ transplantation and all of that, there are barriers to that. Your cells don't want to be in my body and vice versa. So there's some challenges. Classically, a lot of therapies started with autologous. So then the second class that people have talked about for a long time and have always had an eye on, but the science maybe wasn't fully there to support all aspects of it, were allogeneic. And that would be the idea that you take a cell from an individual and you can then give it to another individual or many other individuals. And right there, that already says, well, then from each time you manufacture or make a batch of that, you actually could do it so that you're not making 1 for 1, you're making 1 for 10 or 1 for 100 doses. And that, I think, starts to get us more into where we are with other modalities where you make very large batches of your drug and you're able to then distribute it broadly to people, and that's how you have that really broad societal impact as well. So that's one of the simplest classification systems. On the other hand, then there's the cell types. What cell type, what class or family does it come from? Is it belong to the immune system? Is it part of the bone marrow? Is it a skin-derived cell? But then there can also be the functional. Is it a cell killer? Is it a cell that can secrete molecules? Each of those looks like a different chassis that you could start to build upon. And so the question also could be is, when you start to engineer cells, maybe you want to take 2 of those functions and put them together in a way that they wouldn't normally or naturally occur. And that's also one of the things that really distinguishes, I would say, cell therapies as a medicine, is that we do not have to be restricted to the classic developmental biology lineage of these cells. We might be able to actually take the best parts or the parts we want and assemble them in new ways. And I think that's one of the cool things I think about where the future of the cell therapies as medicines is going.
What are the disease indications that could actually benefit from cell therapies? Is it just oncology or are there more?
No, it's a lot more than just oncology. However, oncology has been the fertile ground where we've been able to see that cell therapies have actually made a really demonstrable impact. And I think in whatever area it initially, or in areas that it can have those impacts, to me that always is exciting, because it's then where else can you go with that? And what are the lessons learned in that one area that you can then start to apply and broaden it into other indications, other cell types and such. And so besides that auto-allo one, the other way, another sort of orthogonal axis is what cell types are you making? What types of therapies can they then be used for? The T cell is, generally speaking, it's a cell killer in a lot of ways, so it helps us in our immune system. So if you have a cell that can eradicate other cell types or targets, cancer is an obvious one, but there are others that we could also start to think about, like fibrosis. We know that comes from cells that, create this fibrous tissue. That's a common problem. The question, though, is do we know how to find the right target on the bad cell type that's responsible? If we do, then theoretically, then you can start to transition that and say, okay, I have a cell and I've learned how to make it kill a cell if I just tell it what the target is. And so that's one of the ways we're starting to see, like CAR T already, is to think about, are there other areas that we could go to? A lot of the other, from the immune or from the oncology side into autoimmune, there's been a major transition in the last 2 to 3 year period with a very logical hypothesis. It's basically that many of the blood cancers that have been eradicated or treated with CAR Ts target B cells. And B cells produce antibodies, and in an autoimmune setting, they're producing antibodies you don't want that are causing that. So people are starting to pay attention. There's some really exciting early clinical data coming out of different parts of the world that is demonstrating, I'll use the term loosely, immune reset. Sometimes that's a contentious term within immunology fields, and I'm not an immunologist. But the simple idea behind that is that if you get rid of the bad B cells and they don't come back, could you permanently help reset the system so that someone who suffers from, say, MS or IBD or some of these diseases, so now you can start to take the learnings from the oncology and apply it directly, targeting the same cell type but just in a different context. And so those sorts of transitions are just starting to happen in the field. My anticipation is you'll see more of that as we get more comfortable and there's more confidence in it. You start to look for new targets, new cell types to go after. It's ways that you can take all of that body of evidence and safety and things that go in, and then start to say, where else could we redirect or redeploy these types of therapies?
Are there examples where people are using cell therapies almost in more of a gain of function, like putting in a missing player?
Exactly. So one of the things that drew me to the field was a long time ago, was this concept that when we started to understand particularly stem cells, was the relation of stem cells to regenerative medicine. So the idea that had started many decades ago was if you had cells that could replace or restore function when we have cells that either lose their function or die or for other reasons are just no longer contributing biologically, then could you bring them back? And if in many cases too, we have, the blood system turns over constantly, our skin turns over constantly. So there's some stem cells in our systems that do that naturally. But one that drew my attention early was the heart. The heart is known as notoriously probably the least regenerative organ. And for some period of time now, people have said, well, if you lose a large part of your heart muscle mass after you've had an infarct, the cardiologist can go in and clear the blockage. So they've taken care of the thing that caused the infarct. But the tissue that got damaged downstream that died because it lost blood for that period of time doesn't come back, ever. And so what you'll always then see is there will be this slow loss of function over time, deterioration of cardiac function if the severity of that initial event is too great. So there's been this ambition for a while. Well, we know what the problem is. You need heart muscle cells, and certain cell types, or stem cell types, I should say, like those that are pluripotent, meaning that they can generate all cell types, they could be used to create muscle. And for a large part of my career, that was one of the things I worked a lot on, was very interested in, and it drew me to that. In the meantime, though, there's been lots of regenerative medicine. And one of the areas that's probably the farthest along, arguably, is actually in the eye. You need a smaller number of cells. You have reasonably accessible, because, surgeons can get to your eye even in a awake patient and do amazing things. And we know in that case also where some of the cells that are lost, exactly where they need to go and what cell type they are.
Because it's so much laminar organization and well-defined. Yeah.
And so very targeted, very focal, accessible. And so the question is, can you make those cells? Now there's been several therapies that have come out for this, and we're seeing now still in phase 1-2, mostly for the clinical trials, but that you're seeing gain of function in individuals that otherwise they would never see it. And that's really, transformative for ophthalmology. I like it too from a scientific, it's like a controlled experiment to some extent. Someone that's generally losing vision in one eye is going to lose vision in the other. It might not be exactly the same, but to think where you have two organs of equal sort of, functionality in the same individual is like the most controlled study you could run.
Yeah.
You have your control inherently right there in the other one. So the patient-to-patient variable, all the other things that can complicate clinical trials, you can see that and measure how well the one eye does. Does it restore or at least not lose function as much as the other eye? And that's how most of those trials have started. And I think what's been exciting is for all those reasons. Can you make the cell? Can you deliver the cell? And do you have a way to measure what it's doing? In those cases, like I said, to go back to cardiac, it's still a great ambition, but you need a lot of cells.
Yeah.
It's a very hard organ to access. And then even there, the functionality, yes, we have cardiac function ones, but to really demonstrate that your medicine and your intervention did that and not other things is very challenging.
Yeah.
So I think, again, similar to the oncology, to establish a basis and then expand, I think for regenerative medicine, targeting ones where you have the best chances of seeing success, leveraging that know-how, many parts which are transferable, and then using that to kind of continue to be able to advance into more and more less treatable diseases at present.
My background is also in epilepsy and looking at like these damaging pathways that occur. And it's very similar to what you were talking about in the heart. And that's even harder to access, but it kind of also gives hope for trying to think maybe hopefully in 25 years, maybe this technology could be used to like regenerate the cells in those pathways that are damaged.
Absolutely. The other thing that's really interesting is the convergence of the two, meaning the immune regulation or modulation of so many of our systems. I think you say that out loud simply, and everyone's like, yeah, we know that now. And I don't think it was always the case. Yeah. And I think that now there's been more attention paid to that cooperativity between our immune system in all of our organs and tissues, that now it's also one of the routes whereby if we really get good at learning how to engineer and control immune cell populations that we can readily deploy into individuals, what else could we access?
Hey, Danielle.
Hey, Wellington.
And look who I found.
What? Steph?
Hi. So Karen's at the bar, but in Italy, so I thought I'd come by and say hi.
Blast from the past. So my question for you is, as always, a definition, twofold. CAR T, what's that? And he talked about immune reset. Why is that controversial?
All right, cool. So CAR T cells, it's an acronym, and it stands for chimeric antigen receptor T cell. And basically what they are these T cells where we've engineered them to express a novel type of receptor so that we can target it against whatever we're going after. Usually we see this a lot in the cancer space. So it's got a specialized receptor that's going to recognize cancer cells so that the T cell can then do the dirty deed of killing the cancer cell. And for your second question, so immune reset kind of gets people on their guard because it's oversimplifying and it kind of actually sounds like it's this hard reboot of the immune system when it really isn't.
So Danielle, Todd talked about two types of cell therapies, autologous and allogeneic. And so how does it work with allogeneic? If you're putting in cells from another person into a person, how does your immune system not reject the cell therapy?
That's a little scary, but it's kind of the same thing with like organ transplants and stuff like that. There's like a series of receptors that you can look at that are on the surface and you figure out if that matches with that patient. So they don't just have like one sitting on the shelf, they have a couple of varieties that would match up with what you have going on in that individual person. You had mentioned how cell therapies might be utilized for immune diseases, oncology, vision, but what are some other areas that they might be utilized?
So I'd say in central nervous system, Parkinson's disease has been one where there's some really encouraging multiple independent trials starting to come out with very clear benefits to patients where They can go off of the drug that they're normally on, that the cell restoration is actually restoring dopamine, and they no longer need the drug that they would normally take. So that's in one of the additional areas. I think as we continue to see it expand is like other metabolic functioning tissues, oftentimes if they get impaired, and again, you can't restore function. Certainly in a ton of excitement around diabetes and being able to restore beta islet function to patients. Again, some challenges we'll have with delivery and manufacturing, but the opportunity that you could restore something that otherwise would never be brought back, I think there's great need for that.
So what are some of the novel tools that you've developed to meet those needs?
So one of the ones that's critical for us is the cell or cell type coming in. If it's a donor cell, it is what it is, but we want to transform it into medicine, so we often have to engineer it. And to engineer it means we have to put stuff inside, molecules that don't exist there naturally. Sometimes they're from other species to kind of do the engineering work for us. And so we have to transiently introduce these into cells, have them transform or change elements. Sometimes it's maybe to knock out a gene so that it will not be expressed. Oftentimes it's to add a component in that they wouldn't naturally have. For example, when we talk about CARs, it's the chimeric antigen receptor that basically is enabling that cell to recognize something that it normally wouldn't. But now that it does, you link that program of recognition to, say, cytotoxicity, and it's going to kill when it sees that target. So to do that means we have to put foreign DNA or RNA in, often DNA, so that it will get integrated into the genome and stay there permanently, at least as long as we want it as a cell to be present in that patient. And then the other thing is the editor itself. So you have all these pieces of it, but you need the often nuclease or a class of something that's like a nuclease that can cut, excise, modify genetic information, but in a very targeted way. So getting those things in, having them hit the right spots, not hit the ones you don't want, and then validating on the back end that all that was done successfully. It's a lot of work, and it's a lot of analytical work as well to prove not just to yourself, but obviously for safety reasons that, things did not go amok. Those editing tools are, I would say, really the key aspect of all of this that over the past, ever since, CRISPR craze and all of that, but it really did kick off that it gave us ability to modify cells in ways that previously was just way more difficult. And that really, I think, has been probably one of the main things that's fueling now cell therapies overall, is just that what is now possible, and it keeps rapidly expanding into other ways that you can customize cells.
A lot of questions that I've heard people ask about these cell therapies is, how are you going to track them? How do you know that they're still in the body? Because they're meant to mimic our cells and be very, not different. So is there any kind of drive in the field to have a uniform way to track them, regardless of how it's being used? Because I feel like that's needed, but how do you deal with it?
Yeah, it's a great point. The short answer is no.
Okay.
And it probably has not gotten enough of the attention yet. I think maybe in part it was, can we even make these things? Can you deploy them in patients? But I think the point you're bringing up now is, that's great. Now, where did they go? What did they do? How long were they there? Because if you're trying to interpret, especially as you get more and more patients, and the results are going to inherently vary, was it because of something that happened in that individual that was different than the 5 others or not? So I do think it's an area that needs more attention. It's on our radar. It is something, too, to think about. There's lots of choices, so which are the ones make them the most sense? If you're engineering a cell already, is there a way to link that into them? You're already going to be making changes. Is there an easy way to do that makes it more readily imageable or traceable, whichever modality we use for that? So that part, I think, is still pretty early, but I think in some areas more than others. Again, in some of the ones like the ophthalmology space, there you have techniques that you don't have to label. You can actually go in and they can tell anatomically, okay, we made a change, and the cells are still there. But as soon as you get into most other organs or tissues or systems, no. And if it's circulating in the blood, yeah, you can sort of detect it. But if you infuse a cell that you want to go to, say, a specific target organ, and it's no longer in the blood, does that mean it's no longer in the body, in the system, and it's gone? Or is it hanging out where you wanted it to go?
Right.
So those kinds of questions have started to come up in clinical trials where it's like, well, we know the persistence of the cell is here for 2 weeks because we see it in the blood. That's fine if you wanted it to only stay in the blood. But in many of these cases, that's maybe not where the mechanism of action is. And so you need to start to think about, how else are you going to actually track these things? So it definitely needs, I'd say, more attention.
Danielle, Todd talked about the difficulty of delivering these therapies. And as we know, we don't generally want things to get into the brain, so we have the blood-brain barrier. So how do we think about this for cell therapies?
That's an interesting thing for like two reasons. One is, for the blood-brain barrier, just as a reminder, that's basically the blood vessels that are in the brain have these really tight junctions. They lack fenestrations, like these little holes that are in them that you have in the rest of the blood vessels in your body, so stuff can like come in and out. So we don't have those in the brain. Instead, we have these really tightly closed doors. And so trying to get a cell across that is like massively harder than something like a biologic, like an antibody, because it's just way bigger. So I don't know how effective it would actually be at getting across. And that's kind of like, taking a step back from cell therapies itself, like one of the things that we're constantly trying to avoid in the brain is like inflammation and stuff like that. And part of what can happen is like if you have immune cells that were able to easily get into the brain, they can start different types of cell signaling, and they would try to recruit other cells, and you get this kind of like runaway train of inflammation. So when you start thinking about utilizing a cell therapy in that space, it might be a little bit tricky, right? One of the things that people look at for cell therapies is if those, engineered cells are able to like proliferate at the site of action. Well, that could be a really huge problem in the brain where it wouldn't be in like a tumor, for example. So I've not seen a lot of literature come out yet about how that's utilized in the neurospace, but I think that there's going to be a couple more engineering tactics that would have to come into play so that you don't have inflammation and too much cell expansion in such a tightly contained and regulated region. So besides just tracking the cells, what are some of the other things that we might want to understand about these therapies once they're in a patient?
Sure. So in the T cell space and all these T cell therapies, one of the key determinants or correlates with a positive outcome is how much expansion of those cells do you see initially upon infusion into a patient. Generally speaking, if you don't see expansion, then it means that they haven't really seen the antigen, they haven't responded to it like they would naturally, and then oftentimes you'll see persistence or just continued growth of a tumor. But by definition, if you're putting a cell type in that has a lot of expansion, that also kind of fulfills at least one of the criteria for what we would generally call a tumor.
Yeah.
So it's this fine line often between you want some, but not too much, and you want it to only be under the conditions that you're really trying to prescribe it for. So when it could go awry, and if it could go awry, and in early trials especially, those concerns about we don't want to give, and this is the example, an autoimmune patient something that could be tumorigenic. In an oncology patient who's already suffering from it, not that you want to give them a tumor, but it's a different clinical context and what you're trying to, and so those risk-benefit estimates or perspective that you have to bring to that. But we don't want to make that mistake and think, we've got this great cellular medicine or drug, and yeah, it is in one context, but it actually could be quite harmful unless you know with a high degree of certainty that it's not going to behave in some aberrant way.
Yeah, I mean, if I'm going to dose an antibody to a person, your body's going to naturally break it down and clear it over time. So there's a natural guardrail that the body does, but putting in a living, breathing cell, we've got to engineer those guardrails.
Yes. Yeah. And actually, it's one of the things I'm personally excited about with the allogeneic space. We've got this little window. We have some ability to have cells from one person persist in a patient, maybe in some cases only 2 to 4 weeks. But if you only need 2 to 4 weeks, then I view it as a good thing.
Right.
You don't want to give it so much immune privilege or evasion that it could stick around forever because you've hidden it. That is a goal for some of the ones like regenerative medicine, where you'd love to give something that restores a cell and it stays there for a long time, at least the duration of the individual. But in some of these other cases, I think it's something that we don't want to over-index, I guess, on the persistence factor without looking at, how long do we really need it? I mean, the other way I'd put it is for most other modalities, do you stay on a drug when you no longer need it? Or do you take it until it fulfills its purpose? And if the biomarkers and clinical indication is that it's no longer needed, you go off the medicine. So I think it's slightly different. I mean, it's than what the initial ambitions were of the field, but I think it's falling in line, and it's again shows where how it will start to align itself like other more mature medicines.
And one of the things that, just not just because it's a hot topic, but I kind of get a view that this might be really helpful. How is AI and machine learning helping to kind of accelerate this? Because it's like there's an infrastructure challenge, but also time and like thinking about all these variables. So how's that playing a role in your field?
So it's early stage for it, but in the areas where I'm seeing it have a major impact. So in stem cell culture, the way that most of us got trained.
Wait, so when you say stem cell culture, you're talking about culture in the cells or the culture of the scientists?
Handling of the cells, culturing of the cells.
Cool. Okay,
but there is a culture to the scientists for sure.
Every single one, yeah.
So its origins has gone through is it's very qualitative in terms of the morphology and metrics of how you get trained. Meaning, you look at a computer screen. You no longer have to look through oculars of a microscope because there are no oculars on most microscopes that you use in cell culture labs. You look at the screen and point and say, okay, yeah, these colonies look good. Well, why do they look good? Oh, well, this edge here, it's nice and condensed, and the morphology nuclei, they look nicely packed. Oh, this spot over here, though, this is a sign it might be starting to differentiate a little bit. You should watch that. So that's how I was trained. That's how people I've worked with have been trained. That whole time, what that implies is there's an algorithm in people's heads that knows what is good and bad. They can even anticipate from enough experience, like which ones will work a certain way or not. And that always drove me nuts because it's a 1-for-1 training. You pass that on the next person, then it goes through their processor, and it's going to be a little different. It's like you get this. So one of the powers is could we use that? And so things like we're working on presently, I know many others as well, is how do we capture enough images of these under these conditions, and we can do the training elements from what we deem good or bad. But there's way more depth in that. There's all kinds, probably thousands of features in each image that are somehow getting processed into what we call very simply good or bad, but there's more definition to it than that. And so that one, from an image-based approach and machine learning and then AI, to not just tell us what it is today, but how it's going to look tomorrow. And is it going to differentiate properly in 2 weeks? That's where it could be the most useful for folks in the lab, because often what will happen is you can have a sense, oh, my culture might be drifting. Do you stop then, or do you wait until it actually crashes? And most people would say, no, they keep going with some hope, at least for a while, that it's going to be okay. How many days do we lose in research time or development time when we could have, if we had known that it was just not going the right way, that stop it then, start a new experiment, start a new batch. In early days where not everything is going to be 100% success rate, it would be great if we knew which ones, with some even likelihood, which ones are more likely going to give us the outcome we want, and which ones are destined for failure. And let's just stop it now.
That's been like the best description of my last 2 weeks.
Danielle, can you explain what Todd means by T-cell expansion and how it doesn't happen if they don't see the antigen?
Yeah, so this is a cool thing about T cells in general. So in reality, you have a whole library of immune cells, meaning that there are these types of cells that once they see something, they will kill it or recruit other cells to kill it. But that whole library, you only have like single copy, and they're designed to wait until something triggers them to then go gangbuster, explode by proliferation, and then that way they can effectively kill something. But it's like having an army on reserve that's only selected when it engages with the right target.
So Danielle, is that part of what Todd is talking about when he says that the cell therapies can persist for time in the body? Are they're hanging out somewhere and then they expand? And so, I mean, that's a big difference between, more traditional molecules that we use for medicine, right?
Oh yeah, absolutely. I feel like there's a lot of work, and maybe I'm just saying that because I'm in this space, but in the biologics world where we're actually injecting functional antibodies into people, those are molecules that once they go in, we can measure pharmacokinetics, and we see that once we introduce them, they only stay in the body for a certain amount of time before they're just naturally cleared. Versus in the cell therapy, they have the opportunity to actually stick around. They're not going to be cleared by the same mechanisms that an antibody would be. It's a living cell, right? So there's like a natural lifetime of that cell that's just inherently different than that of an antibody or just like a complex protein. Let's say that we got past a lot of these hurdles that we're facing in clinical trials. We got some good guys on the table. What would the patient experience look like receiving these cell therapies? How is this different than maybe other courses of action?
If they're an infusible therapy, in theory, you could walk in and have an outpatient and go about your way. Maybe there's something then with a monitorable, device and others that wouldn't require sort of lengthy hospital stays or more invasive types of procedures. At present, most of those are, or in some of these, especially lots of early exploratory clinical trials, there's much to monitor. So you don't want the patient just walking back out on the street. You want to be able to follow what's going on. There may be some early known or at least anticipated potential adverse effects. So you want them where they could get, care immediately if that were to manifest. But in the future, if we work through those and we know what those are, then if we can deploy most or many of these through an infusion-based approach, then it could be possible that people could then get these on even on a repeated basis if it were necessary, and be able to, so you could get infusions of things and maybe you need more than one depending on what type of therapy it is. One of the things that would be interesting to me is we broach from, for example, in the oncology space, from at present really only being able to treat tumors in the blood versus solid tumors. So now can you infuse something that goes to a solid organ, takes up residence there for some period of time, may even expand if necessary there, and delivers on the intended function. So to me then to extrapolate, okay, if I can take that principle, can I take another cell that you would normally have to administer or to an organ, let's say the heart, I'll go back to the heart example, but you no longer have to go through a surgical procedure. Could you engineer the cell to recognize when it gets to the appropriate site and do it through an infusion? So you could actually broach also the delivery aspect of this, because there are cell types that can get to many or all of our organ systems, even some that can penetrate into the central nervous system, into the brain. Again, tissues that are thought to be very inaccessible normally, and in many cases from a safety, you don't really want to go poking around too much. So I think if we understand that and learn from the biology of those cells, how do they do that, and can we transfer those properties to other cell types, then you might be able to have something that could broadly address lots of different therapies in a reasonably safe, minimally invasive kind of procedure.
In 10 years, what are we going to learn by engineering these cells to invade tissues? Again, like I kind of live in the tumor space. I'm super curious what we're going to learn about, the inflammation levels of tumors and how to overcome and bypass that. Even if we don't utilize the cell therapy, I feel like there's stuff that from the engineering that's going to help us figure out how do we kind of tip that over into our favor.
No, definitely. Going back to these immune cells, right? If you can put the right immune cell in some of these cases, like the solid tumor and the way that it takes over and changes the local immune environment, if we understand what's changed, one of the things that's been really interesting but also powerful with some of the cell engineering, you can change normal circuitry. So if a cell sees a inflammatory signal and it is then prone to be sort of shut down in a sense, if you know what that genetic circuit is, you can flip it and say, I don't want you to shut down, I want you to get active, or I want you to secrete an inhibitor so that you're no longer sensitive to that inflammatory molecule or some kind of cytokine that might be shutting down its function. So if we know from the biology what it is, we can engineer solutions for those that could equip a cell to have traits or properties that a host cell doesn't have, and could actually be targeted to overcome what we know about, say, a solid tumor environment and what it is prone to do, and at least attempt, although again, it's, tumors are challenging, but can you outwit elements of it? Can you come up with sort of countermeasures to counteract what they're trying to do, and then use that as an inroads to see, can you get better therapeutic efficacy?
Are people even thinking about that in terms of there's been therapies that, against disease X, and when we look at the patient population, there's a certain percentage just that are not receptive. Maybe there might be even room to utilize the cell therapies to kind of maybe switch over the physiology in that disease area to make them more susceptible. Is that a thing?
It isn't yet, but I agree with your thinking, which is that, yes, are there things that cells could do that could make existing modalities more effective? And it's one of the ways to think about it is that, still go with the mature technologies. One of the things I always say, even as a head of a cell therapy group, so I'm obviously very pro cell therapies, is at the present state and for the near-term future, if there is another solution that is going to give you good efficacy, you should pick it first. The reason being, even if it's in a newer area, because you're going to be able to test that with it. The amount of work that's going to go in to engineer a cell, to get through all the manufacturer safety, all this stuff, it's going to be much longer. So the best use of cell therapies at present is to gain access or to try and expand in areas where other proven modalities have really hit some kind of limit or some kind of challenge that they can't innately overcome. That's, I think, in terms of judiciously choosing where to deploy finite resources in any setting, pick the ones where you also can have impact. There are other ones where maybe we could go with a cell therapy that we have a really good bispecific. The bar is going to be really high. As an engineer, I always say that the best opportunities is when something is working at about 10%. The reason is that means you have this high bar. You almost have like 90% of headroom to try and improve into. But if something's effective like at 90, 95%, you've got a very small space and you could do something in that space and make it better. But if you had a choice between the two, where should you spend your time? I would always err on the side of pick the lower one where you could have impact, have the most sort of do the most good. So I think that's one of the ways to just look at for cell therapies is where is it that we can't today get with some of our other modalities? What would be truly transformative? And let's focus our efforts there and try to find those instances to put our time and effort to.
Where do you think the biggest next breakthrough is going to happen for cell therapies?
It's hard. I think one of the things I'm most excited about to see, and I think that the maturity of the technologies and the confidence in cell therapies is what I call the sense and respond. And that is that, cells are naturally processors with all of this sensory information on their surfaces that equips them to say, where am I, who am I interacting with, and then how to respond. When we learn how to use that for medicines, you can have things that are very much sort of programmable and active only where and when you need them. So right now we're putting cells in and we want them to be active oftentimes from the point we put them in, be ready and prepared to kill cancer cells right away or expand right away. But there could be cases where for safety or other reasons, what we really want is that even greater specificity. And I think that's one of the ways that we could then also reduce side effects, systemic kinds of things. If you have a drug or a medicine that only acts at the point where it's needed, I think you enhance safety and other things quite a bit. And we're getting there with the know-how as well as the biological information. So that's what I'm most excited is that I think that's a whole new area for medicine in general that cell therapy can deliver on that really leverages what cells are prone to do.
Thank you so much for joining today. This is a really interesting and fun talk.
Thank you so much for having me. This is fantastic, and I always love doing this.
This was a super great conversation. I really missed you guys. I'm really happy to be back.
Oh, we missed you too.
There was something that Todd said that I thought was really interesting where, back to your theme of unmet need, he's talking about we don't want to use these cell therapies for everything or where we already have a really good solution or a really good treatment. We want to use this where we haven't been able to make an impact for patients yet. And I thought that was just a really interesting thing in terms of, we don't necessarily want to use the latest tools just because they're the latest tools, but really want to use something where it can make a huge impact and difference.
Totally agree. I mean, that's the thing that's really hard in the oncology space is sometimes I think we take it for granted that our ability to target certain things is actually quite limited. It's not as open as you would think. And having a therapy that could persist and actually infiltrate into like deeper tissues and be able to expand and be more effective is such a, I don't want to say godsend, it's such a crucial addition to our tool repertoire that, anything that gives us an additional opportunity is just, that's the magic we need, I guess.
I don't think I've ever heard somebody be more cautious on our podcast, and I don't know whether to be like totally excited or like maybe this is not quite a mature technology. He literally says, if you have something better that you have now, you should go with that first. What's your take on that? Where are we?
I mean, it is the new kid on the block, right? Like we haven't been using cell therapies as long as we have biologics, for example. And I think like all of us, every scientist who shows up to the bench is constantly thinking about the patient. And if there's an easier, faster way forward, absolutely take that. The longer you take, all of that time is basically equating to the quality of life for patients. And so, you know, in terms of him being really cautious, I think that that's the right way to publicly talk about this new kid on the block, right? There's a lot of work to be done. So I wouldn't be worried. I would see this more of like a call to arms to come in and get to work. We have lots of things to look at. That to me is not daunting. That's exciting. That means we've got really neat things to work on, and there's really great opportunities and potential in this therapy. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stalking cells.